Okay, so now that we started talking about other patterns of inheritance besides Mendelian inheritance, we're going to be looking at some Punnett squares that demonstrate how to perform these crosses in various patterns of inheritance. So you'll need this practice packet, or if you can print that out, that would be great, or you can open it and write on it with Kami, or you could just write your own problems down if you aren't able to do either of those or don't know how to use Kami. What I'm going to do is we're only doing Punnett squares for a few of the examples. It doesn't make sense to do Punnett squares for like a polygenic trait. It's just too complex. So we'll do just 4 of the possible patterns of inheritance. And I'll do 1 video per pattern, so they're nice and short, and you can focus on just that one. I'll do 1 practice problem, and then you can do the rest of the practice problems on your own. And that'll be one of your assignments to submit the remainder practice problems that I don't do for you. Hopefully you have this chart available. If you filled this out while you were watching the instructional video, you'll have the description information that'll be helpful for you. Okay. So we'll start with incomplete dominance. Complete dominance would be what we saw with Mendel. We're going to work on Punnett squares with incomplete dominance. So this first question asked you to describe or define incomplete dominance. So if you remember, incomplete dominance is when we have one allele not mask the recessive, so it's not dominant over the other. So the heterozygous genotype will give us a mixed phenotype. We have one allele that's not dominant over the other. So they both are shown, but in a mixed fashion. So let's look at number 2 as an example. In number 2, we're looking at Japanese 4 o'clock plants. And in this case, the red, or capital R, is incompletely dominant over the white, which is lowercase r, and the heterozygous condition gives us a pink flowering plant. So let's start with our key. Remember that heterozygous gives us the mixed combination, so we're going to actually use a complete genotype in our key instead of just an allele. So big R gives us the red flowering plant. I'm going to do our heterozygous next, which is a pink flowering plant, and our homozygous recessive, which is a white flowering plant. So we'll continue with our problem. For each of the following, construct a Punnett square, give the genotypic and phenotypic ratios. So if we have a red plant and a white plant, we're going to reference our key. Remember, the only way to get red is if we have homozygous red. Remember, the heterozygous will give us pink, not red like we've seen before. We're going to cross that with a white flowering plant, which will be homozygous recessive. Now remember, these are not technically dominant and recessive because one will not dominate over the other. All right, let's do our Punnett square. So we're going to draw our box. We'll cross our red flowering plant, big R, with our white flowering plant, which is little r, and we'll fill in our Punnett square like normal. Notice all of the offspring in the F1 generation are heterozygous, and they are all pink. Remember, we have the mixed phenotype because this is a trait inherited by incomplete dominance. Now, our genotypic and phenotypic ratios will have a little bit of difference now. For the genotypic ratio, we're used to seeing the 3 different genotypes. So we have how many are big R, which is how many are versus how many are big R, or how many are little r. So we have homozygous dominant, heterozygous, homozygous recessive. It will look the same here. So we have none that are homozygous big R. We have all 4 that are heterozygous and none that are homozygous recessive. So that genotype looks the same that we've seen before. Our phenotypic ratio is a little bit different now, and we're used to seeing just 2 numbers, the dominant versus the recessive, but we don't have just 2 phenotypes here. We have 3 phenotypes. If we have 3 colors, we have 3 phenotypes. So now we're going to see how many are red, which we've seen before, looks like they're dominant, how many are pink, and how many are white. So the order here makes sense. Homozygous on the ends, the combination in the middle. So in this case, we have 0 that are red, we have 4 that are pink, and 0 that are white. So our phenotypic ratio has more possible phenotypes recorded, but it also looks a lot like our genotypic ratio. So for the next two problems, part B and part C, I'd like you to do the Punnett squares on your own using the descriptions that are given, and then figure out your genotypic and phenotypic ratios. That will be part of your first assignment for the week, is to complete these problems as well as the other problems in this packet.